There you go. Good morning, everybody. My name is Adam Friedman, and I am with Informatics Group, obviously. Today, I'm going to talk a little bit about and demo some tools for doing enrichment analysis for differentially expressed genes derived from RNA-seq data. I'm going to give you a little background first. So, most people doing bulk RNA-seq work, there's a vast majority of work involves identifying individual differentially expressed genes of interest. And then secondly, for looking for sets of genes that may be, from pathways that are functionally interesting. People often don't know what they think they're going to find. In some cases they do. The analyses I'm going to talk about today are largely based upon, they're not the sort of like shotgun approaches where you throw a whole bunch of different expressed genes at a database and see whether some pathways are rich. This is more focused testing where you are interested in specific pathways. So it's not kitchen sink. But it's important to note that there are features of RNA-seq data that need to be accounted for explicitly rather than just dumping your gene list into a package like Panther, which I've done. I've been guilty of it in the past, but now I am infinitely wiser. Having said that, so this workflow I'm going to show you kind of builds on our previous intro to kind of doing differential expression analysis with Limavoom. In that, we're picking up with the same data set on Drosophila melanogaster. There was a paper published, I think, in 2014, looking at parallel climate adaptation, and so they had in Panama and Maine, they had fly populations. They developed isophemal lines and subjected them to temperature treatments. So you basically high and low temp treatments, and then you have geography, a population level factor. And the idea was their goal was to see if there was any signal of similar adaptation to temperature taking place across different regions. That data was based upon read mapping. We also, in our previous workflow, talked about using pseudo-alignment-based tools like Callisto-based pseudo-alignments to use sleuth for differential expression testing. Later, I'll explain that we can import sleuth results. I won't do it, but I'll be able to explain how that works a little bit. But back to the tools. So what we're going to do is we're going to run through. We're going to quickly race through the steps to get the differential expression, the expression object, the stuff to kind of a recap of what we did last time, a bare bones one, to get to the place where we can do the enrichment analyses, and then we'll do those. So that basically the two things that are important about enrichment analyses with RNA-seq is that expression levels are often correlated between genes, especially in genes of pathways of interest, right? A pathway is operating in concert. We expect that there is going to be some covariance, right? But a lot of tools used for just like naive enrichment analysis don't account for that. And so there is that. And the second issue is that so there's one way to account for that, and things like GSEA, gene set enrichment analysis. Try to manage that by permuting sample labels, right? Because if you permute sample labels to get a p-value, you retain the correlation structure. It's only when you break that correlation structure up that you create problems. The problem is that, when you're swapping sample labels in a traditional standard RNA-seq bulk experiment, you have very few samples. And so, permuting labels 1,000 times makes no sense because you only have so many permutations. And so it's quite possible to get, by chance, p-values of 0 just with a small number of samples. And so two tools were developed separately that were kind of baked into the Limavoom package. One is called Camera and one is called Roast. And that's what we're going to demo today. Camera performs competitive tests. These are similar to things like when you use Panther. You basically test the enrichment for differential expression in your gene set to a background set, right? So you can think of, I had this pathway, these 50 genes in this pathway. I want to compare expression in that 50 genes to all the other genes that we had got expression estimates for. The second tool is ROAST, and this performs what I call our focus tests, which are basically, they're not comparing to a background, they're simply examining the extent to which there's differential expression in that gene set. And these tools are complementary. They both are designed to account for the correlation structure in expression when they're calculating p-values. They do them through different methods. Camera estimates kind of the correlation matrix, between expression, and then it kind of makes adjustments for it. Whereas ROAST uses a rotation method that kind of is a kind of derivative of Monte Carlo based methods, which is, I'm not going to get into the details, but needless to say, they both do what you're supposed to be doing for RNA-seq data. And both of these tools are able to take in multiple gene sets such that you can get basically adjusted p-values to account for the multiple testing. And so we're going to look at 2 gene sets today. When we do enrichment, we'll look at heat shock proteins, which may be important in temperature response, and then MAPK pathway, which are also known for physiological response. So we have 2 gene set files we'll load as well. But anyway, I'm going to basically go through, and the way I'm going to do this so I can talk a little more, since you love to hear me talk, is to basically, I'll click on the embedded links and the markdown, and you can certainly follow along with this rather than copying and pasting or live coding. That seems a little bit, I don't know. I'm not going to do that, but I will explain, and you can see my screen. So we're first, like the original bulk RNA-seq workflow, we have to load all the relevant packages, which we need the DGE expression object, which is part of EdgeR, and then lemma, and StatMod is something that lemma depends on. It's a little puzzling why lemma doesn't just bake it in as a dependency, but so I'm just gonna click on my little RStudio, run the current chunk. Voila, my libraries are loaded. But if I don't hear into my console, it should show that they've all been loaded properly. Then, and this is all like, this is just a recap. So we have this table, which is our sample table. And right, we always need this with a difference expression analysis, because we have to be able to define our samples. And so you see, we have these 12 samples, we have population, we have temperature, and we have basically the way the experiment was set up was there was 3 biological replicates for every population temperature combination for a total of 12. And then we got to open up the expression matrix. And this is basically, for those who are familiar with this or are still fuzzy, this matrix basically, it has each row is a gene, right? And these are the gene labels for, and each column is a sample ID, which is the SRA accessions. I will also say that we can do this for an isoform level matrix as well. There's no difference. You would just need to identify a set of isoforms instead of a set of gene names when you're actually going to do enrichment analyses. So we have the gene expression matrix loaded. Also, as you may or may not recall, lemma is expecting integer counts, but because RSEM was used to derive these estimates, it produced fractional counts because it's accounting for multi-mapping uncertainty, and so we have to just round the matrix. So it's just this round function, and we basically have our matrix. We create the matrix name, our RNA-seq matrix, by rounding the gene data. Voila. Then we also do a filtering. This is very standard, where we filter out rows based upon if very few samples have any expression at all. If we're going to have very low power to detect anything, any differential expression if all the samples are lowly expressed or there's lots of zeros in there. So this is kind of arbitrary, but if there's 12 samples, we're saying let's only pick rows. We create a Boolean filter that basically says, does a row sum, and says, let's take the row sums of this, right, of the counts per million, which is what lemma boom operates on, of the matrix, and we're gonna sum the basically the rows where the CPM levels are greater than 1, and if basically this becomes a Boolean, it's evaluating whether it's greater than or equal to 6. And if we look at just to give you an idea, in case this is still fuzzy, It just basically the gene names and whether it's true or false. You see, this one was obviously false. This gene did not have enough samples, so it'll get filtered out. So we have this Boolean filter, and then we apply the filter to our matrix. We call our filter matrix here, and basically the filter, the rows are selected on the filter, and the comma, when you have a comma in R, in a square bracket with everything after, it says we take all the columns in those rows. And just a little recap, if we do, and then we do dim on CPM filtered matrix, we see we dumped like about almost 5,000 genes had just low expression of those, which is perfectly normal. We then create this digital gene expression object using this DGE list function from edgeR on this filtered matrix. So we're no longer operating now with this kind of like table we've been filtering. We're operating with an actual different kind of expression object. And then again with Lima, we always do some normalization, and we're using TMM normalization. If I'm racing through this, this is simply because this is what we've done before. I'm trying to get us so we can actually do the enrichment analysis. Okay, it takes a minute or two, not even. And then, okay. Now, so we're going to basically do the enrichment analyses on temperature for a two-factor design, because we want to account for population level effects as well. And so, as with our previous workshop, we do this, we create factors from the columns in the table, where population, then we specify the levels for temp. These are two different ways of doing it. You could not set levels if you wanted for population as well. But then we create, this is how we create a design matrix. We do model matrix, and we basically specify, a linear model, right? So this is basically, the matrix is based upon the population plus temperature. If we were doing interaction term, it'd be population star temperature. And this shows you the design, right? So this is our linear model design. And it's important to note this, we'll come back to this, when we're specifying contrast, we can specify by a column number or a label. So we now remember that basically the coefficients in the linear model will be basically, a function of the value for temperature high, right? We're then going to run limavoom to create, and so as I explained in the previous workflow, we're using this voom with quality weights, which is a way of weighting samples by, their presumed, whether their extent that they're maybe outliers or have weird expression patterns, that this is a way to, as long as samples are not just total garbage, you can kind of downweight some samples and not lose replication. It's basically, and like a lot of these difference expression methods, it's trying to calculate a mean variance relationship, which it will use in basically doing some estimates of variance, gene-wise variance. It borrows among genes to calculate variance estimates for specific individual genes. And basically, this by default, it plots out the mean variance trend, which looks pretty standard, and then it shows you the weights on individual samples. These last few steps, we do LM fit, and then we do the empirical Bayes procedure, which is the last step. We don't actually have to do step 12, but I'll just do it so people can just be reminded. Simply, and if we do head all genes, we get this table of our genes, logful change, the average expression across samples, the moderated T statistic for the test, and then a p-value, the adjusted p-value, which is a Benjamin-Hochberg. Anyway, so here we are. We don't even need that, but now the goal is to run the enrichment analysis. So the first thing we're going to do is we're going to use run camera. We want to load our tables, right? We're going to load a table of MapK genes. I got these from the KEGG database. Oh, it headed over here. So these tables have column names of gene ID for them. You could easily give them a column name which would be more informative. Later, I'm going to show you how to just provide names in the enrichment analysis function, so you can actually see the labels when you have more than one gene set. Because if you had them all with gene ID, you get a bunch of outputs and not know which is which, unless you knew ahead of time how many genes belong to each row of the table. So the thing is, the way this works is that we have this list of genes, but there's a handy function that's part of lemma that basically creates row indices for the genes that you're interested in. And then you can supply these indices so it knows which genes are the ones you're being used to test, right? So we're going to create this map k indices running the IDs to indices function. And basically we have to match the map k genes we supply the map k genes as well as the row names it will match to, which in cases it's not, the matrix itself doesn't have a column of gene names. It is the names in the matrix, right? So we do row names, basically supply our R set and the background set of genes, or the overall set that we're looking through. And now we have indices. You see this 1, 10. Oh, right. So there's a bunch, right? I think, and so these are the indices of the genes from the MAPK pathway. Now, so there's 2 ways of running camera. One of them is camera PR, which runs on pre-computed test statistic, such as the moderated t statistics that Libavoom has. And I should also say, just as a reminder, we looked at the design matrix, we want to be reminded, right, what our column names were here, because we want to make sure we specify the right column. Otherwise it takes the entire matrix. But if we look, so now we know temperature high is what we're interested in, so we're going to want to run this on the 3rd column in the table. So when we do this, we call this, here's our results table, and we're piping the camera PR function. And so the fit object has a variable for the t statistics from the differential expression tests. And then we basically, we want the 3rd column, which will be temperature high. And then we supply the MAPK indices. So we supply the test statistic and indices of those statistics that we're looking at for our test set. Now notice how so we have a modestly significant p-value here. There were 88 genes, and the direction of regulation of significant was upregulated. Because we had gene ID as the column name, this is what spits this out, but we know this is our MAPK data, so we know how to interpret this. I will say that there's a default setting for this intergene correlation, right? It's supposed to correct for it, and I'm a little bit mystified by this, but it's the default correlation is very weak, 0.01. And the authors say this is because they picked this statistic, this correlation level, because in this case, camera will rank biologically interpretable sets more highly. This gives a useful compromise between strict error rate control and interpretable gene set rankings. And what's annoying is that you have no options with camera PR. You have to there's no way to change that setting for the correlation statistic. I was surprised about this. I also kind of like go over my head over, don't we expect in some pathways, especially in Drosophila responding to environments, like in our case, there to be stronger correlations. And if you are dealing with stronger correlations in gene sets or have an a priori expectation of that, you certainly want to not be looking at tons of false positives. And so what you may recall if you were in our last workshop is that there's tons of differentially expressed genes in this experiment. Drosophila are very responsive to environment. There's geographic variation and like a very large fraction of genes, so that there's probably going to be just correlations by definition, whether those are individual responses or they are functionally connected. You got to deal with this. And so the way to do that is to use the standard camera function without pre-computed statistics. And what you have to supply are basically, you can supply the expression values, right? And then it looks, and then you specify the contrast which is our temperature high function, and you specify your design matrix, right? So it's evaluating expression, you know, and we, from the Voom weights object, and why do I use this? It's because we use these, the weights, the normalized data that were quality weighted. And I would say this is always important. Like if you're running these tests, run the tests on the same expression values that are used for your differential expression tests. Don't go back and run camera with the raw, unfiltered expression data that went into, that you loaded from a table from RSEM. So anyway, so basically, this is gonna do the estimation and report the correlation coefficient among genes in the pathway, and it will adjust the p-value accordingly. So remember, we had a very significant, well, marginally significant test when we assumed a light correlation, a weak correlation, and look at this. The correlation is really high, right? It's like 0.22 compared to 0.01. And so it's basically saying, when you adjust for that, there is no significant enrichment for DE signals in this MAPK pathway. Another thing that's cool is that you can actually, with either camera package, you can load multiple gene sets and it will provide you an FDR value. So it's kind of like when we do gene level testing, we get a Benjamini-Hochberg corrected p-value, which is listed as, I think it's p-adjust in the lemma output for gene tests. But in this case, it says FDR on the table, but it's basically effectively the same thing. So basically we have to load a heat shock protein table, which is another list of those fly-based genes. We create the indices as before. And then we're gonna run camera. Now we're gonna do something a little bit of magic here, where we're gonna basically run camera, but we're gonna call the index function here with a list, and we're gonna provide this map k equals the indices for gene ID, because we know gene ID is the column, and heat shock equals HSP indices, the gene ID column. The nice way of doing it this way is that the map k and heat shock become the labels in the results table. They become the names of those vectors in this list of vectors we supply. We supply the same design. We also supply the contrast as well. And then we do not supply intergene correlation because we're basically telling camera to estimate it. So in this case, neither pathway has significant enrichment, and the FDR value is well above what we call significant. So anyway, this shows you that you need to be careful about thinking about intergene correlations, because if you don't, you might get a whole bunch of positive signals. Something I'm going to note, as I said before, is it's entirely possible to run camera on expression data from another tool. Way you can do this is you can basically load, all you need is a vector of test statistics, right? So if you take the test statistic for the differential expression testing from Sleuth, I think that's a T statistic as well. You simply load it like you load the T statistics, or you can use log fold change values. There's some other options. You need to pick what makes the most sense, but you can load those in and do the same kind of operations. You just have to know in the Sleuth object where to find that tabular information. And it's there. You certainly also could, another way to do this is if you didn't want to use lemma Voom, you could bring in an expression matrix from another tool, right? From another tool. You could take Callisto if you wanted to do lemma testing with Callisto data and not use Sleuth. You could use a package called TX import to build an expression matrix from the pseudo counts derived from Callisto. There's a number of ways to get here if you have some problem with lemma itself. The one last thing we're gonna do is targeted analysis with ROAST. And this is gonna yield quite a different result since it's not looking at the background, and I'll talk about that in a second. But so the mRoast function is a roast function for calling multiple gene sets. And we do it very similar. We simply call mRoast like we would, we call camera, or we bring in our expression levels. We bring in this index where we named a list of named vectors where map-k, again, heat shock are the names. Our design, our contrast, our adjustment method is, we say, is BH. I think it's by default it is. Oh, one last thing about ROAST is you have to pick a set statistic. There is a number of statistics that you can choose from. And I have some text down here. I'll just summarize it. Basically, which is the appropriate statistic depends upon the extent to which you think there's the fraction of DE genes in the gene set. Mean 50 is a good balance between false discovery and power, where you can detect as many, if only 25% of genes are DE, or you can do a reasonable job. You would use it, if you use the mean, just mean instead of mean 50, mean is used when you expect that almost all the genes will be DE. And then there's a MSQ set statistic, which is used when you expect that very few. You know, it's shown to have power when you have as few as 1% of genes. So again, it requires a little bit of user thought before just running these, unlike if you just dump stuff into an online enrichment database. And so I should just run this. And we see is, look at this. We have our labeled lists. We have very significant enrichments, apparently relative for temperature. So you know, there's downregulation relative to the high temperature treatment, and then the reciprocal of that, the opposite of that for map-k. Now, one final note here is that, okay, why is this test yielding such different results? As I said earlier, there's a lot of differential expression going on in this experiment. And so if there's a lot of differential expression going on, it may be harder to determine whether there is differential expression that's more than normal, right? So ROAST isn't comparing against the background, simply saying, is there a kind of a general DE signal in this data set, regardless of what's going on in the rest of the genes that are detected as being expressed in the sample. So again, there's some thought as to which test makes more sense. Don't simply pick ROAST because you think it's more likely to give you a significant result. But again, it becomes more challenging, you know, if you expect high levels of DE across the board, to basically have a background you can actually distinguish your differential expression from. I think that is all I've got to say on this. So I'm going to stop recording.